All right, good morning, everybody. Bring up everything on chat. Actually, who was here today? Okay. Get stuff sorted. Right, so where we ended up on Monday, remember the day of the week, is finishing up with the overview of control of gene expression in eukaryotes. So essentially, kind of how do we get from a universal set of instructions in a multicellular organism to considerable differences, right, in terms of physiology, right? So here's some more. I'm pretty sure these are confocal images as well, by looks of it. They're really sharp. So this is a neuron, probably in vitro would be my guess in cell culture. And these are keratinocytes, right? So those are the cells that are involved in hair and nail and other kind of extrusion type processes. And clearly they have extraordinarily different morphology, right? These are actually two different markers. Let's blow these up a bit because these are really pretty pictures. Promise not to get too distracted, despite my self-confessed microscope nerdism. So the red is microtubulin, right? The same protein that you encountered in cell division is what the spindle fibers are made out of, which pull apart the chromosomes. And this is actin, right? Which is in us. Well, I mean, it's half of muscle, essentially, actin and myosin. Here it's actually used as a structural filament. Anyway, so microtubules are involved in essentially transport within the cell, like kind of cellular highways, essentially. And actin is often involved in kind of maintaining structure. Don't know who that is. And actually, the really fun thing about growth cones, right? So this is how neurons extend their processes to join up with other neurons or their targets, like muscle, for example. And those growth cones kind of spread out like so. It's kind of like a splayed hand almost, lots of fingers like you can see here. And depending on what signals they receive, actually, this is a great example of kind of post-translational function, right? Depending on what signals that growth cone receives, either repulsive or attractive, that skeleton collapses. And so the growth cone continues in a particular direction. If it wanders off, it collapses and keeps going in the same direction. And so that's how you end up joining together all of your millions of neurons to make the right connections. Anyway, same proteins, now in very different cells, right? So these are, I think the word is squamous, right? Which basically means flat and stuck together. And now you can see the same proteins, right? Now we have actin along the periphery of the cell. These almost look like plant cells, right? They're not, but they look like it. And now we have microtubulin in just kind of diffused around the inside of the cell, right? Same instruction set, very different results or outcome. All right. So what I'm going to do, or we're going to start doing now, right, pretty much until the end of the course, depending on how much time we have for kind of fun stuff like transgenics and the like. But between now and then, I'm going to go through each of those different levels, right? So all these different come on, steps that you can see here: chromatin state, control of transcription, control of RNA processing, control of translation, degradation, and so on and so forth, right? I'll be going through those in order, essentially, right? So we're going to start with DNA, and then we're going to end up with protein post-translational modifications, and that's going to be all the way down here. So anyway, as you can see, there's a fair bit of stuff to cover. Okay, so first we're going to talk about chromatin state, and really that's the kind of catch-all term for that is epigenetics, right? So epigenetics are changes to your DNA that don't change the sequence, but change how that information is used, right? I'm going to write that down. Even though you've got a definition up there, this is a slightly different one saying kind of the same thing, different way. So it's modification of DNA that doesn't change the sequence. And DNA in this sense, don't forget, in a normal eukaryotic cell, DNA is always complexed with proteins, right? It's always in as chromatin, right? It's never just bare DNA. So when we say modification of DNA, really we're talking about modification of chromatin. Right, I'll just add that. And it affects gene expression and it's heritable. Heritable? There you go. And so this is actually kind of a crazy thing if you think about it, because, you know, including myself, we've really been taught that inheritance is all about DNA, right? The sequence of DNA, mutations, alleles, and how those are inherited. That's still all true. But there are also a series of markers, epigenetic markers or modifications that you inherited from your parents, actually both sides, in a non-Mendelian fashion, right? Oh, sorry, I got to tell this story. Some of you probably heard it already. One of the kind of coolest parts about this, and there's been a couple of studies about this, not very many, but a couple, is that for the ladies amongst you, right, you're the oocyte that made you, right, that, you know, was the gamete that your mother joined with the gamete of your father, right, to create you as an organism. That oocyte was made while your mother was in her mother. So you make your oocyte, as a woman, obviously, you make your oocytes when you're a fetus, right, before birth. And so at a particular point in embryonic or fetal development, you have three individuals essentially in one place. The grandmother, the mother, which is the fetus, and the future daughter, which is the oocyte, the eggs that are being made, right? Magic? I don't know. Oocytes are, it's actually kind of crazy because they are arrested at, metaphase 1. And so all of the oocytes in guys, it's different, you know, just make them continually as they kind of get used up. But in women, you make them all before birth, and then you kind of cull them, which seems a little bit, you know, harsh. Sometimes some cells in your body are constantly being renewed. Skin cells, for example. Others are not. The neurons that you have at birth are the ones that you'll die with, at whatever age that might be. So yeah, but that's also why rates of meiotic non-disjunction that lead to things like Down syndrome increase with age, because, you know, if you're 20 and you conceive, that oocyte's been around for 20 years. If you're 40, it's been around for 40 years, and there's a risk that it kind of slipped through that M-phase checkpoint inappropriately, essentially. So that's why the Down syndrome instance curve increases with age. Anyway, so it's really cool because that basically means that whatever the grandmother experiences, you know, that can be imprinted on her daughter and her granddaughter all at the same time, which is kind of mind-blowing, to be honest. Doesn't really affect guys though. Sorry, guys, you're not that special. Anyway, so there are a bunch of different epigenetic modifications. There are ways of remodeling chromatin by things called chromatin remodeling complexes. There are, which we'll talk about a bit because they're kind of neat and interesting, but we'll also spend a bunch of time on histones and their modifications, right? So remember, histones are what DNA is wrapped around to form chromatin. And also modifications of DNA, right? Which is something called methylation, right? So there's actually a lot going on here. This is actually an astonishingly complex subject, particularly number 2 here, right? There's an awful lot going on and an awful lot we don't actually understand. So essentially, heterochromatin or compact chromatin equals transcriptional repression. Genes are off, right? And it's generally believed that that's the normal state of affairs, right? And so the reason that transcription is off is that these bits of DNA, right, that are important for beginning transcription, right? So the Tata box binding protein site, which brings in RNA polymerase, the transcriptional activator protein site, which helps activate the transcription complex, right? If these aren't physically available, transcription cannot start. And they can be unavailable for a couple of different ways, right? They could be inside like a tangled complex of chromatin, right? So the proteins physically kind of can't get in there, like TBP or any activated proteins can't get into that DNA sequence to even bind to it, right? So that's one way compact chromatin shuts things down. Another way is that this DNA in heterochromatin is tightly wound around the histone proteins. So even if the Tata box binding protein could get to that DNA is not in a position where it can stick to it. And so if those sequences are unavailable for either of those two reasons, transcription does not happen. Right, so this is, again, coming back to the concept that in general, transcription in eukaryotes is off by default. Obviously, there's different reasons for that, but this is one of them. So essentially one thing that you can do is, aside from anything to do with the protein kind of nature, right, modification of those, you can just remove them, right? So if you remove the histone proteins from that stretch of DNA, now that DNA is potentially available, right, for proteins to bind to it, transcriptional proteins. You could also kind of shuffle them along, right? So that it's like an abacus, you just kind of like push them to one side, right? Now that bit of DNA is free and available. Or you could rotate the histones, so it's like having a thread on a bobbin or an anchor chain around a, what was it, capstan? And so if you just rotate these histone complexes, that DNA may end up between the nucleosomes rather than wrapped, part of them. And so these are three different things that are all carried out by chromatin remodeling complexes. And that takes energy, right? So they use ATP to do so, right? And depending on how these work and what the outcome is, you could both positively or negatively regulate transcription, right? If the action of the CRCs is to make these sequences more available, you're going to get an increase in transcription. If it's to make them less available, then obviously you get the opposite. So here's a good example. Let me blow this up a touch because this is a little bit on the tiny side. This is a good example of that rotation of the nucleosomes, right? So these are big proteins, protein complexes, right? Made up of a lot of different protein subunits, essentially. And they're often directed to a particular site, like go here, because of modifications of the histones themselves. Right, so there's an awful lot of interplay between all of these different things. And that's what makes it such an astonishingly complex subject, right? Because there are lots of different levels and all of these different things talk to each other, right? So anyway, once this CRC's been recruited, now it can rotate the DNA on the nucleosomes so that the DNA that was kind of bound to the histones is now free in the middle, right? And that means that those transcriptional proteins can bind, right? And so, for example, we've got the transcriptional activator protein, we've got TBP, that brings in RNA polymerase. And now, once RNA polymerase is there and activated, it kind of acts almost like a snowplow. Some of you might never seen a snowplow. I don't know, like the thing on the front of trains in old Western films. And it will just push those nucleosomes off the DNA as the RNA polymerase goes down and transcribes that gene. And they just kind of reassemble after it. You can also just kick them off, right? So in using these CRCs and ATP, you can physically remove an octamer, one of the 8 histone units that makes up a nucleosome. And so that, again, same outcome, makes that DNA available for other proteins to bind to it. And that's really the main deal. That's kind of what runs through all of this. Apart from DNA modification, that's kind of different. But in terms of chromatin remodeling and histone modifications, it's all about how accessible is that DNA. Can things get to it to begin transcription or not? If they cannot, no gene expression, right? Now I don't have one of the sliders, never mind. So something that's actually received an awful lot of attention, and I've got a somewhat superficial understanding of all of this. I mean, I get all of the kind of principles, but some of the details are just straight over my head. So essentially, there's two opposing modifications of histones, and it's all about changing the charge of the histone protein, right? So kind of going back again, way earlier in the course, actually, what's the charge? And actually, this is something that we've covered in lab a whole bunch of times. What's the charge of DNA? What charge does DNA have? Awesome, it's negative. Thank you, all you lot. Okay, so can you change the charge of DNA? Think about why DNA is negatively charged. Can you change that? Maybe, no, not really. Good thinking about it though, for sure. And the reason is it's part of the structure of DNA, right? So DNA has a negative charge because of the phosphate groups in the backbone. So you can't change that, right? Because it's an inherent property. So if DNA is negatively charged, what charge do you think histones can have? Yeah, exactly. And so if you want histones to bind tightly to DNA, those histones are going to need to have a positive charge, right? Now, again, you can't change the charge of DNA, but now you can change the charge of the histones, right? And so if you wanted DNA to kind of flow up and be looser, what are you going to do to that positive charge of histones? What has to happen to it? Yeah, more negative, actually just neutral, just getting rid of that positive charge is going to release that attraction, right? So, I mean, negative charge would kind of be even better, essentially, but it's a little bit harder to do. It's actually a lot easier just to get rid of the positive charge. And so histone modifications are really all about the charge of the histone protein, right? And that dictates how tightly the DNA is bound to it. And that in turn dictates how available the DNA is for all these other things to access it. Okay. And so those two different modifications are called methylation and acetylation. I'm going to type these out. So, and actually, let's kind of just go back here for a wee second. So you see these little, they look like little men or women stuck upside down in the sand for some reason. I don't know why they're drawn like that. But you see these little bobbly bits sticking out the side of those nucleosomes. Those are the histone tails. So they're part of the histone protein, but they kind of flap around outside of the nucleosome, right? And so those are what will be modified, right? Because they're accessible, right? You can actually have other proteins can come in and change their properties. So histones, what would you say? Naturally, maybe have a positive charge. And that's due to the lysines, lysine residues on their tails. Let me see if I've got that. Oh, no, don't want that. I want, no, not that one. Bear with me. Here we go. No, didn't want that. Migration. Where are we at? Here we go. That's what I wanted. All right, so where's lysine? Lysine. Blow up. lysine, positively charged, right? So the positive charge comes from specific amino acid residues in the histones. Those are lysines. And so these are the residues that we need to neutralize if we're to affect the charge of the histones and how tightly they bind to DNA or DNA binds to them. All right, so this is kind of neat because it gets back to some fairly fundamental properties that we covered a little while ago. Let's go back here. Okay, so, and here, that's what I want. So, to neutralize, oh, okay, and let's just kind of carry this a little bit further through. Positive charge, get back there, attracts the negatively charged DNA, equals tightly bound, inaccessible DNA. So to change that, to release the DNA, those lysines are modified by the addition of an acetyl. I can never remember how Americans pronounce. How do you pronounce that word? Acetyl? Acetyl? Anybody? Help an expat out. I need to learn the local lingo. Acetyl. Ooh, okay. Thank you, Kaylin. That's a help. Okay, so the addition of an acetyl group. Sorry. Removes that positive charge. So acetylation equals transcription. I mean, obviously, there are other things that have to happen for transcription to occur. It's not an automatic deal. It simply means that that DNA is now available for transcription to occur if all the other conditions line up correctly, right? All the other proteins are present. Now, you can stop that from happening by, and I'm not entirely sure where these methyl groups get added. I should look this up. By acetylation, by methylation or methylation. And that's a I don't know if that's to lysine 2. Actually, let's just look it up. I ain't not knowing these questions, answers to these questions. Ah, there you go. Now I know, right? So that can also happen on lysines and also on arginines. It's arginine. Oh, arginine is also positive. Look at that. There you go. Learn something new every day, as they say. Cool. So, aha. And so typically, that equals repression of transcription. Not always, because it's very context dependent, but typically, right? So you have two opposing modifications, right? Acetylation, gene expression, methylation, gene repression, by and large, right? So you can kind of switch between those two, right? Depending on what is going on and what other signals are present that dictate chromatin structure, right? Also, these modifications interplay with other chromatin modifiers. Modifiers spelled that way, I'm not really sure. Right, so the methylation or acetylation state can actually recruit other proteins, right? Like these chromatin remodeling complexes, and tell them to do something, right? So if these histones get acetylated, which is pro-transcription, that may help recruit these remodeling complexes, which now rotate the nucleosomes, so that not only is that DNA kind of looser, but is also now functionally available too, and vice versa, right? So this is just, I mean, it's really hard to get across how complex this is, right? And there's actually something called the histone code. We'll get to some pictures in a second. And that's basically the tally of lysines and arginines that are methylated or acetylated, right? And so you often see this written as, actually it's almost like code, right? So H3K4, for example, right? K is the letter for arginine, H stands for histone, so that's histone 3 modified at lysine 4, for example. And there's a whole bunch of these lysines and arginines in those tails, right? So, and that particular code depends on the context, right? So it is astonishingly complex. And so here's some nice pictures. So you can see, let's blow this up a wee bit. Right, you can see how these tails actually go quite some way outside of the nucleosome, right? So here's the DNA wrapped around these 8 histone proteins, but their tails are accessible outside of that, right? And these tails are what dictate how tightly bound or not that DNA is. It's kind of like they either kind of like scrunch in or, you know, relax out. And there's 8 different histone proteins, right? So there's and in pairs. So there's going to be 2 H3s, 2 H4s, 2 H2Bs, 2 H2As, right? And each and individual one of these has multiple residues that can be modified. And so you can see how just astonishingly complex this can get, right? And so essentially, in just general kind of broad strokes, methylated histones equals heterochromatin. And acetylated histones equals euchromatin. Right, so this is kind of building it back up to that bigger picture of chromatin state. And again, that's all about the charge of those histone tails. And so you can actually see here, it looks a little bit like, I don't know, like some weird creature out of Half-Life. That's actually for you, Stephanie, a reference. You probably get that. Not sure anybody else does. Played that many, many, many years ago. And excellent. I knew you'd get that. And when these tails are acetylated, they're just kind of like, floating around in the breeze, right? So they're not tightening the DNA. So the DNA doesn't fall off, right? It doesn't unwind. Those nucleosomes are still intact, right? You can see that they still kind of maintain their functional integrity, but that DNA is now much more available for things to bind to it, right? So you can actually see how something can now kind of grab onto that DNA because it's physically free. And so the crazy thing is as well is that these histone modifications are passed down through cell division. And so, and what's really even crazier is that the DNA in your gametes, sperm or oocytes, has some of those histone modifications. And so it's almost like not only are you giving your child, an instruction set, right, the DNA, all the different genes and alleles and variations and so on and so forth, but you're also giving them, in a sense, a kind of guide to how to read that instruction set, right? Pay attention to this one, don't pay attention to that one, right? And so that's going to affect the physiology, right? The development, the phenotypes of that offspring, in addition to all the information encoded by the DNA, right? So it's this extra level of, complexity. And again, that's why Mendelian genetics is like that much of human genetics. Because not only do we have multiple genes controlling a certain trait, different alleles interacting in different ways and so on and so forth, right? All that other non-Mendelian genetics we talked about. But on top of all of that, we have these histone and DNA modifications, which are not encoded in the DNA itself. So you can see why, you are truly different from your parents, right? It's not just the DNA. And so there's definitely a memory. And actually this memory tends to last, as far as we can tell, and it's really hard to look at humans about this, obviously, for obvious reasons. Typically it dissipates after about 3 generations. So the information that I'm given, basically gave to my daughters, good information, one hopes, right? That information is kind of going to dissipate in their children, and most likely it's going to be negligible in their children, right? So my great-grandchildren, great-grandchildren are going to have obviously some part of my DNA in terms of my information, but my histone modifications, my chromatin modifications are likely to be, irrelevant in terms of, who they are. It's really cool. And actually, in many ways, I just love talking about this stuff because it's just super neat. You can't change your DNA, right? From generation to generation. You can't change your DNA, right? Change occurs randomly by mutation, right? So you have no kind of say over what your kiddies are getting, right? It's just random. It's random what alleles they get. It's random if there are mutations there, all random. So if you're in the middle of a famine, drought, or something nasty, right? You want your kids to be best prepared as possible, right? And so not only do you do things as a parent, but you also kind of tell your offspring genetically, ooh, when you pop out, son, daughter, things are going to be a little bit shitty, right? So physiologically prepare for that. So it's like kind of giving your next generation a bit of a heads up as to the conditions that they're going to experience when they're born. Isn't that just nuts? It's just so much to biology. It's just cool. Go on and on and on. Anyway, so that's histone modifications. And the reason, and you can now see why I spend, well, it's almost a whole class period on this stuff, right? Because it is just so fascinating. It's not just fascinating in like who you are, it's fascinating in terms of who you are relative to your parents and your grandparents. You know, so for example, my nan kind of, you know, obviously was in London during World War II, getting bombed and stuff. My mum wasn't in utero then, but after the war, you know, there's a lot of, you know, restrictions and stuff. Yeah, who knows? Maybe that's why I'm such a hoarder. Never throw anything away. Always might be useful. Okay, so in addition to protein modifications, you can also methylate or modify DNA, right? So this does not change the sequence of DNA. That's really important to emphasize. Your DNA sequence stays the same. Its physical properties change. And this is commonly associated with transcriptional silencing, right? Actually, much the same as methylation of histones is involved in transcriptional repression. Methylation of DNA has the same outcome. And it does so largely because it, well, one, it physically changes how the DNA looks. So transcription factors, transcriptional activator proteins, all that kind of stuff, they recognize a particular shape, charge, sequence of DNA. And so when that DNA is methylated, that looks subtly different. And so those proteins are less likely to bind. Also, other proteins which shut down transcription may only recognize methylated DNA, right? So when they see that change, they're like, oh, and they go and shut things down. And actually, it's really interesting that, so these, they're called CPG islands. Let's see if we've got something. Yeah. These CPG islands, and those are essentially sites of DNA methylation, are often found in regulatory promoters. Right, so the actual part of the gene where proteins bind to turn on or turn off transcription, you often find these groups of cysteines, right? These CPG islands, right? And often in cancer, the promoters of cell cycle regulators, and essentially, I can't remember the correct word, anti-cancer. There's actually a proper word for that, just can't remember off the top of my head. Are heavily promoted, are heavily methylated. And so that's kind of an extra dimension to, you know, cancer biology. Is it's not just about kind of turning on things that should be off, but it's also about turning off things that should be on, right? And so proteins that are involved in blocking uncontrolled cell division tend to be transcriptionally silenced in cancers by DNA methylation. And so there's this constant kind of switch back and forth in state. Actually, it's the same for histones too, right? And so So histones are modified by two groups of enzymes. Histone acetyl transferases, or HATs, add acetyl groups equals gene expression. Histone deacetylases, which are called HDACs, and histone methylases, or methyltransferases, actually, HMTs, equals gene silencing. And you have the same for DNA methylation. You have DNA methyltransferases. Actually, let's do it in the same order. Demethylases. Methylases. These aren't easy words to spell. Yeah. Those remove methyl groups equals gene expression. And then DNA methyltransferases or DNMTs add methyl groups equals gene silencing. So you have these two kind of groups battling each other, essentially. On the one side, you have histone acetyltransferases and DNA demethylases, right? Those are pro-gene expression. On the other side, you have histone deacetylases, histone methylases, and DNA methyltransferases. There's a lot of words there. Those are gene silencing. And so the kind of the ratio or balance of all of those different modifications dictates how available that DNA is for transcription. And they also talk to each other. Don't need to look at that quite so much. So, for example, we have a methylated cytosine. What do they call them? Cysteines. You numbnuts, man. Sorry. Better change that. Oh, actually, I'll change that in. Haha, I can go back and change that after class. Make myself a note to do that. Because obviously cysteines are amino acids. You don't find those in DNA. Okay, so as you can see here, we've got a methylated cytosine, C, right? This is presumably in, let's say, a promoter. That recruits other proteins, right? Whatever MECP2 stands for. But it recognizes that particular shape of DNA, right? And that then recruits a histone deacetylase, or an HDAC, right? Which removes that acetyl group from the lysine from on that histone tail, right? This in turn recruits a histone methyltransferase, which adds a methyl group onto that histone. So you can see that one change leads to another, and now you can really shut down gene expression in a particular area of the chromosome, right? So these can reinforce each other. And here's another good example of this is I thought this was from maybe there's a different example somewhere. I thought it was from lizards. But anyway, the state of one part of the DNA promotes the modification of the histones, which again reinforces that gene silencing, and vice versa too, right? Works in the other direction as well. And now you can actually see that histone code, right? So this is histone 3 modified at lysine 9, for example. Okay, so that's enough for today. There's a lot going on here, right? So just keep in mind the general picture of accessible, transcribed DNA, inaccessible, silenced DNA, right? And the different things which kind of basically moves from one to the other and vice versa. All right, everybody. Yeah, so I'll see you either in lab today or I'll see you on Friday. And don't forget, if you have lab not today or not this week, your lab's going to be after Thanksgiving. Cool. Have a lovely day, everybody. I shall see you all soon.